All right, guys, bang, bang. Super excited to have this conversation with Joe here. So thanks so much for coming on and doing this.
It's a pleasure, Anthony. Great to be here.
Yeah. Let's just get started with your background. Obviously, you guys are working on some very cutting edge medical and technology related products, but what did you do beforehand to really get started down this path?
Well, my background to understand how I got to this point to, build Quantgene together with my amazing team is that I come from a very medical and bioscience family. So I was born to, my parents were both bioscientists, very passionate microbiology and other life science related things. Not in business, just science. And yeah, I grew up in a lab basically with my dad and learned about PCR machines when I was 2 without really understanding anything, but I kind of saw it. And then when it came down to choosing my, college course, what I would study, I actually wanted to get out of that. I thought like medicine, I've seen that enough for 19 years of my life. I like it. It's cool, but I don't know what else to learn here. And so I basically decided, let's become an economist. I was very interested in business and also economics. And so that led me down a kind of more the business path, went into kind of strategy consulting, then mergers and acquisitions and a little finance, then quantitative finance. And I found my passion for everything quantitative, complex systems, but especially fuzzy, probabilistic systems, which are important in finance and in venture capital. And then it came full circle when a family member who's also a doctor and bioscientist asked me for advice on a big math and statistics problem in genomics for cancer related things. And that was in 2014. And I really got sucked into that. I was at that time working on a kind of quantitative venture capital framework with a slight focus also on life sciences on the investment side. But that project kind of really triggered my interest because the deeper I dug into that question, how can you profile DNA, not fragments, but whole genomes and kind of DNAs from isolated potential tumor cells. How could you determine if something is a tumor cell if you just look at the DNA? Which seemed like a trivial question back then, but the more I dug into it, the more I realized this is a very complicated question. And I asked a bunch of friends at Berkeley and Stanford and all kinds of institutions where I knew they know a lot of things. And none of them could answer the question. And then we had some good ideas and they said, well, that's definitely not something anyone has done so far. And we kind of solved a very important math statistics question in genomics. And that triggered basically the foundation of the creation of Quantgene, because I realized, well, if we can solve that problem and no one has solved it, we can detect cancer at early stages in the blood. Because then we had the tools to basically identify specific locations on the genome necessary to detect cancer comprehensively in the blood. And so that's the whole thing.
So maybe let's go deep into the science here, because I think a lot of people listening, they recognize that they don't understand the science nearly as well as you do. So help us understand when you're talking about DNA, like just what is that to get started with? And then we can maybe talk about how certain cells are mutated or kind of die through disease and how disease really kind of runs through the body and what that does to the bloodstream and to those DNA cells.
Sure. So as a first step, I think it's always important to see what cancer detection and cancer is today, right? It's an enormous problem. 600,000 people die every single year in the United States alone of cancer. 1.6 million get diagnosed with cancer. Each of us has a 40% lifetime chance of getting cancer. That's pretty severe. And over 10% chance of dying of cancer. So it's probably one of the largest risks each of us has. And early detection of cancer is a huge game changer. If you look at survival statistics, the difference between, for example, breast cancer detected in stage 1 versus stage 4 is very massive. 99% of women have a 5-year plus survival at stage 1, 9% survival at stage 4. So it's a massive difference, what your chances are, because if it's detected early, you just go into a hospital, get surgery, and you're cancer free in many cases. Whereas if it's late stage, it's like it's terrible. It's an ordeal that goes on years and years of chemotherapy and very bad outcomes. So you wanted to detect it early. Now, genomics is something that was not used or is still not really used in cancer detection. It is something where we do hereditary testing more and more, which means we look at your healthy genome, right? So all your cells have a genome, a DNA, and that is kind of the building blocks of life are proteins, and the proteins are encoded in the DNA. So the DNA is being read out, turned into proteins, and we have like roughly 20,000 proteins in the human body, different ones. And that determines who you are. Now, that is called hereditary, genetics. So all your cells have the same DNA. We can look at that DNA very easily nowadays and then tell you if you have certain predispositions, right? If you have certain variants that other people don't have, you might be more protected or less protected against certain cancers. So that's the first step of genetics into cancer, that we can tell you if you have, for example, BRCA1 variants, specific ones, then as a woman, your chance of breast cancer is much, much higher, that you get that over your lifetime, because something in your cell and metabolism is less likely to protect you.
And how do we find those variances, right? So like we know that now if you have that variance, you have a higher probability of getting breast cancer for a woman, for example, but how are those discovered?
You mean how we scientifically discover the initial insight that there is a problem, or how do we find it in your body?
The initial insight that is an indicator.
Yeah, so there are multiple ways of doing that. There's a mechanistic way where you basically identify a variant, and then you try to find out what that actually does, right? So you can see, okay, if that variant is on the DNA, how does the protein change, right? And you can say, well, if the protein changes, and that protein, for example, is a tumor suppressor gene, like TP53, and it's being changed, then it's a loss of function effect, for example, then the protein stops working. And if that protein is designed to kill a cell if it becomes a tumor cell, right, like a guard that is in the cell waiting for something to go wrong, and that guard becomes disabled, then we know, okay, that will probably lead to cancer. There are lots of other examples, like there are tons of things that you can do. That's one way of figuring it out. Another way is population health studies, where you have basically association studies. So you look at millions or thousands of people, and you see, well, in the subgroup of people who have that variant, breast cancer is 100 times more prevalent. Like often it's not 100, it's 10 times or something. And that then it's more, correlation versus causation. No one knows exactly what's going on, but you derive from that, well, probably bad news if you have that variant. We don't know exactly what it does. But you're in that new bucket, and we see that people in that bucket have 100, 300, 1,000% more chance of developing a disease. So you should be cautious and be on the watch out.
Got it. And really what this gets at is something I find fascinating personally, which is just, we really don't understand the human body that well, right? We understand a lot, but it is such a complex machine that we're still learning things every single day. And so we're kind of doing the best we can with the information we have, right?
And that's exactly why we built Quantgene and Serenity, our kind of patient-facing brand. I think there's a big misconception about science and medicine, and multiple misconceptions. But one of the biggest one is that we know a lot. Like, first of all, that's always a dumb assumption. We don't even know, like we don't know a lot about anything. Physics or chemistry, this is all like, we always scraping the surface if we are lucky. But in biology, it's especially extreme, I think, when you are in that field. Like every single time you go below the surface and you say, oh, this, causes cancer or something. And then you dig deeper into the protein and the protein change. The number of proteins we actually understand what they do in cancer is probably maximum a handful, probably more 1 or 3, like KRAS or, TP53 or something. And even there, it's very unclear. All the details are extremely unclear. And then you dig one layer deeper and you have only questions and no answers. And, every good scientist would tell you, well, we simply don't know. Like, you have to run a bunch of experiments. Once you run the experiments, you probably still don't know. And you only have some statistical or probabilistic insight. And I think that's very important in medicine. It's very different from, for example, building rockets, right? Anything that's kind of artificially created by humans, we have a much better understanding of. If you build a computer or a rocket, you have a pretty comprehensive understanding because you build it. Everything that's naturally occurring, you have just no clue. Like when you look at the body, if you're lucky, you know, like 0.01% of what's actually going on. And it's very clear, you know, the deeper you dig, there are just vast areas of unknowns.
Yeah. And so would it be fair to say that in a lot of the work you guys are doing, obviously is one, trying to solve some of this, but two, just in the broader science and health industry, is that we're really focused on just trying to understand what's going on, right? Right. The solutions only come after we understand. And so that is really kind of the base layer. And a lot of this biotechnology and things that are being seen as breakthroughs, really it's just identifying something that's been going on in humans for, you know, thousands of years. We're just now starting to understand given the technologies that we have.
Absolutely. And I think there is a huge paradigm shift going on in medicine right now, very slowly going on. So that's why we created a company to accelerate that. But the paradigm shift is the medicine of the past and the current medicine. The way of doing medicine is very static, right? You go to med school, you learn a bunch of things and you think you know something, which is a big mistake. And I think the new paradigm in medicine that, you know, Serenity and Quantgene stand for is that it's a much more dynamic system that is an equation of basically three things. Data, you need a lot of data. You need intelligence, you need to make sense of the data. And then action or systems, right? You need to then take some form of action and have some kind of infrastructure to do treatments or more diagnostics. That's not how medicine works right now. Medicine right now is static knowledge. You're like, if that, then do that. Whereas, you know, I give you one example. If you do a conventional blood test, PSA, for example, for prostate-specific antigen, you get one number. It says, oh, 7. And they say, oh, it's above the threshold. I might have a problem, maybe not. When we run a test on you, especially the somatic mutational test for cancer detection, which we didn't get into yet, that's a different thing. We're not looking at your healthy genome. We get 6 billion data points. So we get 6 billion individual nucleotide reads out of that sample. And then we have a very complicated cloud-based, you know, software and AI system that makes sense of that data. And we are building Bayesian layers on top of that to take in all additional information around you. And that system isn't perfect, but it gives you an idea, you know, what we are dealing with here. That system changes every week, like on a deeper level, because it learns from patients and studies. It gives us literally 6 billion times more data points than a normal sample. And it's operated on a different medical paradigm. And the medical paradigm is more data is always better and more intelligence. And maybe you, as someone who's in tech and business, you think like, why is that like a thing? Like, isn't that obvious? Like, of course, more data and more. But in medicine, you know, the majority of physicians would say, no, that's not true. More data is always often bad, because more data means I may have to make decisions that I don't want to make. Because if that test comes back positive, I have to do some things according to standard of care. Therefore, I would prefer not to do the test. And for non-medical professionals, it sounds absurd, especially for tech and finance, like how is this makes any sense? But it's about standard procedures, it's about liabilities. If I don't see it, I can't do anything wrong. And that sounds ridiculous maybe to you and many listeners, but it's literally what most doctors would tell you. It's like, no, if that's a young patient, don't do that test. Because if it comes back positive, I have to do all these things, and that would be wrong to do because it's a young patient. And then you would argue, but if you know it's wrong to do, why do you do it? It's like, well, because it's standard procedure and I don't want to get into trouble if, you know, so let's just not measure anything.
It's absolutely wild how the healthcare system works.
It is wild, and it's especially wild because you meet so many dedicated people who are high IQ, super trained and knowledgeable that do all these stupid things and know they're stupid and say, I know it's stupid, that's why I don't want to do it. So it's this absurd system where we have so much talent and intelligence that is kind of, in chains. They can't do what they should and want to do. And that's why it's so important to break out and build companies that don't have to adhere to these, you know, standard procedures, but can really strongly innovate with the patient interest in mind.
Yeah. And so let's talk a little bit about deep genomic sequencing, right? Because I think that's a term that, at least genomic sequencing, people have heard before. They, everything from high level, just like what that is, all the way to they get to things around genomic editing and kind of all the scary stuff in most people's minds. So maybe you can just start off with just like, what is deep genomic sequencing? And then we can start to talk about, like, once that's done, all of the different applications of that information.
Yes, and that's exactly where the rubber hits the road. So what I explained before, hereditary testing, that is the old school sequencing that still hasn't reached most of medicine, by the way. So old school, I mean, it's 5 years ago. It's still only penetrated probably 5% of medicine. So we need to do that. That's very important. But what that does is it looks at your healthy genome. And to do that on a technical level, you're sequencing something with a depth of 10 to 30x. So what does it mean? It means if you have a DNA strand and you have a specific location, you want to take 10 DNAs and look at specifically that location. And then you build an average and say it's a T, 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 G, A, T, right? So on the same location across 10 of these. And then you say, well, an average is a T. So you have a T, right? So it accounts for some sequencing errors. So you do it 10 times, maybe you do it 30 times. That's how you normally sequence your cells. You basically sample a random 10 to 30 DNA copies and look at the same location 30 times and tell you the average and say, well, Anthony, you have a T here. And that makes you regular or it makes you different, whatever it is. Now, what deep genomics is, and Quantgene is the global leader in precision sequencing. So we have the most precise sequencing technology that can do the following. If you have a blood sample and you have, let's say, 2,000 copies of DNA in that blood sample, of cell-free DNA. So 2,000 copies that stem from 2,000 different cells in your body that died and shed their DNA into the blood. Our technology can make sure with a very high level of confidence that we can investigate every single copy. So instead of picking 30 randomly out and tell you what the average is, we can tell you, well, we sequenced all 2,000 individual copies. And for each of these copies, we can say, this is a T, this is the T, this is a T, but here's a G, and here's a T, and so on. And what that allows you to do, that's a total game changer. It allows you to say, if there's any single copy in there from a cell that carried a somatic mutation. So a somatic mutation is a mutation that is not normal for your body. That's not your healthy DNA. It is something where a body cell that you have is mutated, has changed. Very different from a hereditary variant. So it might be that if you have blue eyes or green eyes or something, you have a G where other people have a T on that location, but all your cells have that. That's why it's you. Whereas what we are doing is we want to identify of all the DNA, cell-free DNA in a sample, is there a single copy that stems from a single tumor cell, right? And that gives you a level of precision that is completely paradigm shifting, because before in cancer detection, you have protein tests. They need literally millions or billions of proteins in that sample to see any kind of delta, right? If you have a PSA of 7 versus 1, I can't tell you the exact number, but we are talking about millions to billions of actual PSA proteins you need to have in there to see that difference.
Would it be fair to say, as you're looking at the blood of an individual, would it be fair to say that you're grabbing the DNA cells out of that blood, and when you're looking, if you're only using a sample, so let's say 10 or 30 or 50 or whatever the number is, you could actually miss one of the mutated cells because it doesn't end up being in your random sample. So by actually sequencing all of them, you're not only, one, becoming more accurate, but two, it sounds like this precision technology you have actually lets you test every single one of them, right? So it's kind of the precision leads to higher degrees of accuracy.
Exactly. So it's very simple. The math is simple. If you have a tumor in your body, early stage, right? That tumor at stage 1 has 100 to 200 million cells. That's a stage 1 tumor. That's somewhere in your body. Out of these cells, a certain number dies every day. Actually, a very significant number, right? So if you have 200 million, at least 2 to 3 million actually turn over per day. So they shed their DNA into your blood. If we take 20 milliliters of your blood, that's roughly 0.4% of your blood. So it's not non-trivial, right? It's nearly 0.5% of your entire blood if you take 2 big tubes. And so you can ask the question, if you have, let's say, to be conservative, 1 million of these tumor cells die every single day and shed their DNA into the blood. Of course, it gets digested, so you have to divide it by 24 hours and then see how much is actually circulating at every minute. But you still, if you do the math, you still get to a point where it's very likely that you have 10, 20, 30 circulating tumor DNA copies in one tube. So for us, but you have thousands and thousands of other cells in the tube of cell-free DNA that messes this up. So if you would randomly pick out 10 copies, that's not enough. You are very likely to miss anything in there if it's at that concentration. So you have to make sure that you look at everything in that tube. And that is something, even 5 years ago, that sounded like complete science fiction. We, 5 years ago, already anticipated where sequencing will be now. So we knew it's definitely possible. So we started developing everything so we can do it now. But 10 years ago, I think 95% of scientists would have said that's complete science fiction. That's like a warp drive or something.
Why did they believe that though? Right. So they said, hey, that's science fiction, but was it because they couldn't actually identify all of the cells in the bloodstream? Was it something around their ability to scale the testing of each cell? Like why did they actually think that that was not possible?
Because back 10 years ago, you need basically 3 things to really make that happen. Well, 4 things. You need some improvements, or you need some quantum leap insight advances in chemistry or in strategy, how you sequence. So it was just completely not possible 10 years ago. But then people found some cool things out about how you can actually barcode DNA, so to reduce errors in sequencing. That's one important thing. That you needed an invention that we couldn't have made, like someone came up with it randomly, and without that it's not possible. But it's also a quantitative infrastructure problem. You need sequencing technologies that are thousands of times more efficient than 10 years ago, and we just happen to have them now, thanks to Illumina and some other companies. You need massive computing power that 10 years ago, most bioscientists would have said that's just not possible to ever get there. And guess what? Thanks, we have massive improvements in computing power. We are reading like 200 gigabytes in the cloud, in a cloud system per sample, as opposed to 1 bit, right? If you have a PSA test and it says 7, no, you need a byte, I think, for that. So you need 1 byte, and we have 200 gigabyte files. So just that delta, you need multiple improvements on multiple fronts that are very massive. And so, even in 2015, when we did the math on that, the costs for 1 sample based on the sequencing technology back then would have been roughly $1 million per sample.
Per sample?
Yes. And wow. Today, we are far below 1,000.
Yeah. And so help me understand, now that you're able to test every single cell that you find in the blood and do it with an increased amount of accuracy, what does that allow you to identify or kind of do that for me as a patient or an individual, I either 1, couldn't get access to before from an information standpoint, or 2, can you tell me, hey, you've got a higher probability, or you actually have cancer before any other test can identify it? Like, how do you put this into application now that you have the technology to do it?
So very different from hereditary testing that just tells you what your probability is of getting something over your lifetime. Somatic testing tells you if you currently have a tumor, which is, of course, vastly more actionable. So what it does is it creates very complex, deep genomics patterns. So what a pattern means is we look at, multiple tens of thousands of locations that we predetermined before. So we know if you look at all of them, we have a very good chance of intercepting any kind of tumor variant across 15 different cancers. So for each of these locations, we then get what's called a frequency, right? Because it's not yes or no. It's like how many mutated fragments do we find and how many non-mutated fragments. So we see at that specific location, we have 0.1% carry a cancer-associated mutation in your blood. At the next location, 0.56% carry something. So that gives you a two-dimensional pattern, right? A deep genomics two-dimensional pattern. And these patterns code for diseases. Of course, the question is for what diseases and how do they code for it? And that's the big statistics and research question. So what we did is we are running very large patient trials. We have a 10,000 patient trial going on right now, 15 different cancer types, and a control cohort, so of non-diagnosed patients. And what, you have to build machine learning around that. That is also hard to build because you have to do many things differently from self-driving cars or, recommendation engines. It's just a totally different problem. You have to build very different things. And we run your pattern against the historic data set. So, right, we have your unique pattern and we run that against all the other patterns like pancreatic cancer, early stage, late stage, colorectal cancer, early stage, late stage, and so on. And control patients that don't have cancer but are smokers, patients that are not smokers, not cancer but are obese. And so we basically run that against all these different cohorts and get a match back. And that match tells us, well, Anthony looks most like X and second most like Y. And then you have a Bayesian layer above that basically says, well, now we know the genomics match. Now we also take into consideration your age, your gender, other background information we have to put additional probability layers on top of it. And then we get the final call that says, well, considering all these things, the system thinks it's most likely nothing, hopefully. Or it's most likely red flag for early stage colon cancer, which doesn't mean you have colon cancer, but it means, well, there is something now to watch out and take potentially some action. And what's very important for us is you have to not just develop technology here, you have to think about medical practice, because you can't just give that to a patient and say, okay, good luck. You have to say, okay, what do we do now? And that creates all kinds of liabilities, regulatory issues, all kinds of things. So what we are building is a system that solves the entire equation for the patient. So we say, well, based on all these things, we think you should do a stool test, even though normally you were not supposed to do that, because you have enough risk here that we say, if we do a stool test, like poop in a bucket, with some genomics test on top of it, we can rule out colon cancer with a high probability, or rule in, right, whatever it is.
Yeah. And so what I'm taking away from this is there's a couple of different steps here, right? So the first is what I'll call just collection of the raw materials. So you have to actually get the blood or whatever it is. Then there's an actual scientific process that is identifying the cells and kind of having that precision technology or science that you talked about. And then now what you're talking about is this third step, which is really just data analytics, right? It's very kind of high level. It's very large data sets, but it sounds like machine learning type problem where you ultimately can say, hey, we have confidence that we collected the samples from the people that we think we did. We've got confidence that we were able to use our precision technology to get accurate identification, kind of the raw data that we have we're feeding to the machine is accurate. And then if those machine learning algorithms and kind of computers are structured correctly, they should be smarter than the average doctor, right? And come back and say, here is exactly the path that this person is on based on the genomic sequencing. And then you guys almost are going to build like a recommendation engine after that then says you should go get this test or you should go do X, Y, or Z because of the machine learning has matched you to these other people. And that's where we know how to make that recommendation. Do I have that kind of right?
Yeah, I think you got very close. There are two more steps that make this whole thing work after that. The next step is clinical algorithms. And that is something you cannot do through a machine learning system. You have to do this. You need a lot of people involved. You need to apply what's called a Delphi method. So you have to ask a lot of experts for their opinion. What happens under all these circumstances, right? For all these different cancers, what downstream clinical diagnostics should we apply? And here you cross the line from science into medicine, which is a very different thing. So medicine is all about who agrees, who are other people saying the same thing, because you have to get to a consensus among experts, otherwise you're screwed, right? You need liability out of the way, you need to implement that. So it is a very different field than just science. The scientist can just say, well, I think this is the best solution because here's my statistics. In clinical reality, you have to say, but who are the people who agree with that? Because it's a much more holistic problem. And then the last step, so fourth is clinical algorithms that this whole thing needs to work with, which is very complex. That's one of the main reasons why smart tech guys who go into biotech often fail, because they lack the understanding of what goes beyond science and engineering, what goes into this weird clinical space, this authority and doctors and experts and legal and compliance. But you have to solve that, otherwise you don't have a product. And the final step is, of course, the business model. Like, who actually pays for that? And that, again, sounds maybe trivial to most tech and finance guys. Like, why is this? I mean, if you save people's lives, they just pay for it. Guess what? Not in medicine. So, we changed that fundamentally, but it gets insanely complicated. How do you convince a insurance bureaucrat that he likes that? And if you show that you save people, like, sorry to be so frank, but they don't care, right? That's not their job. You have to show them that they save money, and you have to show them that you save money immediately or quickly over time, and then, things get insanely complicated, and that kills also most of the innovation. So if you don't solve 4 and 5, all your amazing tech is not going to work because you can't finance it. And so this is like now I gave you the full picture and complexity, why it's absolutely non-trivial to truly understand your business model, to innovate on that front, and to be highly synced with the medical and clinical reality of things. And then nail the tech, and then you're getting somewhere.
Yeah, it's absolutely nuts. And I guess part of that clinical side, when I hear the word clinical, I think of all sorts of different tests that can be run. With this genomic sequencing stuff, is this only being done on humans? Was it previously done on animals? And then that's where the technology kind of was perfected, and now it's being brought to the human side? Like what is the relationship there in some of that testing to non-human kind of clinical trials, or whatever you could do there?
Well, the problem is, or the advantage of genomic sequencing is, you don't have to do a lot of animal trials because there's no risk in just collecting the evidence, right? So that was very human driven from the outset on, Craig Venter and the Human Genome Project. So you can sequence, a lot, and you don't, without any kind of approval, because if you do clinical trials and you just sequence, no one gets hurt as long as there's no feedback loop. So it's very different. The actual sequencing and developing sequencing technologies, you're free to test this on humans because you're not putting anyone at risk, especially if there's no feedback of information. Once it gets into cancer screening, of course the game completely changes, and that's the problem. That's what's holding the liquid biopsy industry back. So what we discuss is called liquid biopsy, because it's liquid and kind of a biopsy. And that's where we innovated a lot to actually get out of that problem. And get medical advancements and technologies to our members much faster than we normally would be able to do that if we just go through Medicare and commercial insurance.
Got it. And then I guess as you get better and better at this, right? Because part of the machine learning algorithms that are actually analyzing all of this sequencing, they should improve over time, right? It's kind of the beauty of machine learning. Help me understand. Is that something where, let's say I'm testing for a variation that we believe could lead to a higher propensity for cancer, right? Can I use the advances and the accuracy that we've determined on that test in those machine learning algorithms to then apply to a different type of test? Like, is there some shared knowledge or shared analysis that can be moved from test to test? Or is each one of these tests, when you go to start them, they're really starting from scratch, right? So you have to rewrite the algorithms and kind of start all over again without any of that shared knowledge.
I mean, I'm super excited about these things because it's probably the most challenging and rewarding data science problems I've ever seen. Well, there are some in finance that are also pretty immediately rewarding, but I mean, in a human sense. And you can view it as like, in a way, if you take a step back, it's like any data problem, right? If you want to determine something, if you want to figure out if a certain real estate, offering is like likely to increase in value over time, you could build a crazy machine learning thing around it, like how many people look at it, where it's listed, what the current price is, what the stock market does. Like, you could feed all that information. And how does the stock market data relate to that real estate as opposed to other real estate listings around it? Well, no one knows. These are two different tests, right? You combine these test sets, and if you have sufficient data sets, you can make them work together. And that's, if we test your hereditary, if you understand your hereditary variance, and we understand your somatic variance, the somatic variants are also like a time-based, kind of on a time series, because we do that every year. Whereas your hereditary, we don't have to do every year because it's not going to change. So you have very different types of data sets, and so you have to learn how to make them compatible. But each data set adds a significant amount of insight and dimension to the problem. I think there are two major challenges in medical data sets. The first one is they are often very heterogeneous. So if you have 1,000 or 10,000 patients, the probability that you have the same data sets for these 10,000 is like 0, unless you do a super clean clinical trial that costs you an immense amount of money. If this is commercial patients, then I might have your hereditary genetics, but not the next one, next person's. I might have your hereditary and your somatic and good clinical records, but for the next person, I might only have hereditary and some records. So when you build machine learning systems, it's very important to account for that, right? That you sample, the comprehensiveness of data is nearly impossible to maintain. And just cutting it down to the data dimensions that are comprehensive is kind of stupid, because you're losing so much information. So you need much more fuzzy, systems that allow you to recognize much more things. And you also need to embrace more, man-machine hybrids, where you have experts and analysts sitting in front of these things and use the machine learning more as a decision support system and have these mutual interactions. That's very important in medicine. And then the ultimate problem, of course, from a business perspective, is you need to close the business model loop in order to even get that information, right? You need to figure out, how do I actually make money and create a business model here? And that's very, very hard. And I'm very happy we solved it the way we solved, because for most companies, that means you have to just do very rigid clinical trials over a decade that costs you hundreds of millions of dollars and are super inflexible. And in the end, you have bad data sets because you missed out on the most important dimension. We see this with a bunch of our peers that just don't have the right data sets.
How do you guys make money? What is the business model that you figured out that works?
So we made a very bold step that some love and some hate. And we said, I'm an economist by trade, right? So I love to look into a system, and understand, why is this not working? And medical innovation is simply not working if you see what's possible in theory and what's happening. There's a huge delta between what could happen based on all the tech we have and what does happen in medicine. It's ridiculous. My conviction, after looking at this for like over a decade now, is it all boils down not to the FDA, not to the government, not to regulations. That's smaller obstacles. It boils down to the payer problem. If you have socialized medicine, which we effectively have, or bureaucratized medicine, which means if I protect your life, as with a new technology, the value I'm generating is your value, because you value your life and your health and your family's health. But you're not the payer, right? Or at least not normally. We change that. So if I have to create value for people but get paid by other people, I will stop creating value for you because you're not paying me. I start to creating value for the insurance company or Medicare, which is the same thing. So what do they want? They want to cut costs. They want to collect your premium and pay as little as possible, right? And they want to do so on a short time horizon. They're not interested if I saved them money in 10 years because I prevented you from having stage 4 cancer and saved, hundreds of thousands on your cancer treatment. They ask me, if I give you that test now, within 3 years, can you recoup the test? And of course I can't. It's not possible. Because, if you test 100 people, you detect 2 cancers. For these 2, you save a lot of money, maybe. But maybe if you wouldn't detect it, maybe they would have had symptoms only 4 years down the road. So they're off the table for insurance companies. So, once we address only the payer's interest, the patient is out of the picture. No one is going to innovate anything that saves you a life or protect you, because you're not paying and the payer is not interested in that. It sounds very brutal, but it's also very obvious for everyone who knows business. And so what we said is, if you want to build a company that stands for true medical progress, you can only do that if you have a self-payer model. You have to get paid by the people you protect. Because if you let other people pay for it, you're not going to protect them anymore. And so what we develop is can we develop something that is affordable for most people, but still provides margins for us, that makes us a real business. And so we are at a roughly $200 a month membership fee for a once a year test. So it's $2,400 a year for that deep somatic test. It's an $800 one-off fee for the hereditary testing, but that's a once in a life thing you do. Then we have an intelligence layer. Once we do your genetics testing, that you continuously get updates on any drugs that are incompatible with your genome, that pose a risk to you. Any other insights we have on an ongoing level, that's $20 per month ongoing. So it's all affordable. But over time, of course, we want to make this cheaper. And over time, this allows us to actually build the patient base and the clinical evidence to get insurance more and more on board, because we're developing that evidence over time, but not by raising $1 billion and do a 200,000 patient study over 10 years. So it's a little controversial to just go for self-payers, but we see 2 things. The market trend is on our side, because a lot of patients understand that you have to do that if you want to get actually advanced medicine. And second, we also see that it's the only way of doing it. As I said, as an economist, I have no illusions about incentives in business. If you get paid, you just want to save costs, you're not going to save people. It's very simple.
It's funny how the same capitalistic tendencies that the finance industry has exist in medicine as well, right?
I mean, yeah, to think that giant publicly traded companies, pharma, insurance, and so on, don't think about the bottom line is a little delusional. And once you understand the logics of it's not that they are bad people, but you know how it works. You have to report your quarterly profits. You have to show that you grow your business. Same with pharma. If your business is to sell chemotherapy drugs and you make $161 billion a year as an industry, are you really incentivized to cure cancer with vitamin C? I'm not saying that this works, but I'm just saying, let's imagine vitamin C would work. That would be the worst disaster in the history of pharma if that actually works. They would lose $160 billion a year. Yeah. And so the incentives are a little flawed, right? So no one is I'm not saying anyone is actively trying to not cure it, but if I pitch any big, pharma company with, I have this great idea, it costs only $10 to cure cancer if it works. I need $10 million to do this trial, for a stage 1 trial. I mean, you can be guaranteed that no one is going to finance it, because I say that that's an absurd proposition to us.
Yeah, it's pretty crazy. The other thing around genome sequencing that I think a lot of people have heard of, but they probably don't understand, is when you get into all of the gene editing, right? So whether it's at the individual cell level or kind of this DNA editing, maybe talk a little bit about, it's kind of hard for me to see that world happening if we don't actually understand first, right? We kind of have to understand the genes and the DNA and kind of what's going on before you can even think about editing. But there's obviously technologies out there that people kind of have heard at a high level. And how do you think about that as maybe kind of a long-term path that the medical community is likely to pursue, whether it's just for, curing diseases and kind of direct, health applications versus like, hey, I want to change the color of my baby's eyes type stuff?
Yeah. Well, I think the extreme depth of potential knowledge and the lack thereof in biology sounds like a daunting problem, right? So, we don't understand anything. How can we even edit? But I think this immense amount of depth and complexity and the lack of knowledge opens the door also to genius, right? You can be very genius by saying like, maybe we don't have to know all these things. Maybe we can hack it. Like, for example, if you know a mutation does some bad stuff, but you have no idea what, you just know in the outcome is bad, but we cannot figure out what it actually does. Maybe you don't need to figure that out. Maybe you just remove that mutation through gene editing. And maybe you cut it out and replace it with a healthy piece of DNA. So the whole CRISPR revolution, I think it's, often things get overhyped, but CRISPR is a little bit like the internet. Everyone said the internet, oh God, it's going to change our lives forever. Everything is going to change. And everyone's like, oh my God, so obnoxious, these people, they're overhyping this. But it was just true. I mean, it is changing everything. And CRISPR is very similar because CRISPR allows you to, it's kind of crazy when you think about it. It allows you to, in a very targeted way, take any specific segment of the DNA and replace it with any desired fragment you want to put in there to the nucleotide. You have 3.3 billion nucleotides on your DNA. They can, to the single nucleotide, say, okay, at that specific location, cut it out, inject that sequence. And they can do it in living organisms. They can basically inject it into you, and it would kind of, disperse across your whole body in theory and replace it in all your cells, which is extremely freaky because it actually works, right? So you could actually change your eye color in theory while you're like living, or even change all kinds of things. So that is extremely freaky. Of course, there's an enormous amount of things that can go wrong. You're probably going to die if you want to change your eye color because some stuff is going to happen that no one was considering. So I wouldn't test it out at home. But in theory, it's definitely possible. And so we know a lot of things, but compared to what we should know, it's basically zero. But that doesn't mean you can't do absolutely amazing or shocking things. And of course, we are very focused on disease and keeping people healthy and, not let them age too fast. There are many things you could do, but of course you need very, very deep clinical trials for that to prove safety, especially if you start editing genomes in living humans. It's more a regulatory issue. I mean, scientifically, it's definitely extremely possible, and not even that, you can do it today.
And where do people get, so like, let's say I want to take one of those nucleotides and I want to replace it, right? So I've identified that one is a bad one, whatever, for whatever reason. Where do I get the replacement? Is that something that I take from somewhere else in the body? Is that something that's like, quote unquote, lab grown? Where does that replacement one come from in that scenario?
So here, and these are great questions, because if you go into biotech and you will learn about these things, it's absolutely stunning, how hard certain things are where you think that can't be that hard, and how easy certain things are where you think this must be completely impossible. And then someone's like, no, it costs $5, go on the website and you get it. And it's like stunning. And this is one of these things. You can literally say, here's my sequence, TTAACGACGA or something, right? That I don't know where you get this from, but whatever, you do your research and say, I want this little part of the protein change, these amino acids. And so I have, these 24 nucleotide sequence that I want to inject here. You can literally go on the website. I'm not going to promote any specific companies, but you can Google that quickly, copy, paste your TTACG things in there, take out your credit card, and they send it to you 2 weeks later.
No way.
In a little tube.
No way.
And then you have them, and they are clean. You have a few billion of these little sequences in there, and they're all that sequence.
And what are people doing with those today? Are they like injecting them?
No, there's a huge amount of stuff where this is used. We do this every day. We order them every day. So, you need them in sequencing, for example, for PCR reactions. So they're called, these nucleotide sequences are hugely, used all across the board. They are, for example, used as primers. So we can use them to basically have a little sequence that reflects another sequence on the DNA. And if you put that into a mix and do some stuff with it, heat it back up and down and put polymerase in it, this enzyme, then these primers will anneal, like they will align to the complement on the DNA, and then start reading that out, like whatever comes after. So you can basically say, I want to read out whatever is on the DNA after that sequence. That's actually sequencing. So you say, okay, if you want to read out that specific part of the DNA, what you have to do is a primer that you synthesize that sits here, and it then aligns here, and then the polymerase enzyme comes here and starts complementing the rest of the DNA. And then that gets put on a sequencing machine, and you read it out, and you see, oh yeah, this is my primer, I recognize it, that's what I designed. And then what comes after is the sequence that I was looking at. So I can see if you actually have a T there or a G.
It's kind of similar to like in the movies when they like clap really loud so they can sync the audio and the video, right? It is kind of very similar.
Yeah, it's the same thing, kind of. Yeah.
Yeah. Before we go to wrap up, you have been a Bitcoin proponent and have a pretty cool story about Chamath, who's obviously come on the podcast. Maybe tell a little bit just about like how you discovered Bitcoin, because I think it was just pretty interesting that.
You had some very cool people on the podcast. I'm a big fan of Chamath. I think he's, of course, super smart, but he's also outspoken. And he is what a smart investor always should be, and a smart entrepreneur. He has these different perspectives. He thinks in first principles about certain things of the world and then applies them. Same with Kathy from ARC, right? Also amazing. So I met Chamath in Harvard Business School. There was an event. I didn't go to Harvard. I was just at the conference. And I think in 2014. And back then I had heard of Bitcoin, but not really. I mean, that is 6 years ago. And he gave this very, passionate pitch about Bitcoin and how it's going to change everything. And he made this, I don't know, he just totally convinced me to get into Bitcoin just with a little bit of money. So I bought a bunch of Bitcoins at $800 because it's how I do my investing, right? I analyze stuff, but I'm also really listening to entrepreneurs and smart investors where I, on a meta level, see, okay, this guy knows something, he's smart. The way he thinks resonates with me. It's the way I think, but he thinks about different things. So I'm listening to him on these different things. And that paid off pretty well. So thanks so much.
Yeah, that's an awesome story. Before we wrap up, I always ask 2 questions, and then you get to ask me one to finish it up. But what is the most important book that you've ever read?
Well, I actually forgot the title of the book. It was in German. I'm from Germany, so. But that book, what was actually the title? I know the cover. It was black and had some weird orange shape on it. I read this when I was 10 or 11 or something. And that book was basically taking you on a journey. I forgot, you have these, there are these famous guys in the US who do this similar thing. It takes you on the journey, on the evolutionary journey from the Big Bang to us today. And I remember one specific thing in the book that was kind of a game changer in my mind. And that was the theory of life, right? So the most exciting thing for me is not how monkeys turn into humans. It's like trivial. I mean, a monkey is basically a human. Or like even a fish turns into whatever, a rat, kind of. That's like, fine, I get it. So, but the question is, how does this all start? How does something dead turn into life? And in this book, he described that in a way that was so logical, that was shocking, how dead stuff turned into life. That basically, if you have a bunch of molecules, randomly assembled proteins, right, in the soup, in the beginning soup thing, right, in the sea, and you just put energy and light on it, so they start changing. They mutate randomly, not as life, but as just dead matter, as proteins. Like, oh yeah, there's a little light here, and it changes like that. At some point, randomly in that soup, a specific protein, or substance or molecule will change in a way that it will align to other molecules and replicate itself. And that's just a pure question of statistics. To that point, it's like, of course, at some point, that's just a random trait of the thing. But at the second where it does that, guess what happens to the soup? These molecules will be much more, prevalent than all other molecules because they're the only ones who replicate. And that's the first step to life. That's what viruses do, for example. So viruses are not alive, in case people didn't know. It's not a life form. And so, that was for me stunning because it was exactly that magic steps, like, ooh, there's dead stuff, stones, and there's life when you look at humans and stones. But when you narrow it down to this one thing, what is actually the main trait of life, to understand, well, the only thing that it does, it replicates. And, and that is that's pure math, statistics, chemistry. It will happen inevitably because whatever replicates will be more of than the other stuff. And then it's just a race who replicates more effectively, who can eat other stuff, who can develop DNA. Then you're off to the races. But that was such a revelation because it's a purely mathematic explanation why life is inevitable.
I remember the first time, and I forget the name of the book as well, but I read about telomeres, and they were basically talking about the replication. And you're just like, wow, this is a whole world that I don't understand. I don't know if I want to learn more because it's scary, but at the same time, it helps you understand life for sure. You spend a lot of time thinking about human life, obviously, and kind of what humans are made of and DNA sequencing and all of that. What about aliens? Are you a believer in aliens, not believer? What's the thoughts there?
Well, the Fermi paradox is one of my favorite topics. Like why, if you do the math on it, it's kind of obvious we should have millions of alien civilizations, even higher developed civilizations. And why didn't we meet them? It's one of them, are they just lingering out around Earth and don't make contact because we are too primitive, or what's the thing? And I have my own theory there, but I think it must be true that there are tons of civilizations. But it also is very likely that they don't go beyond a certain civilizational development stage, which allows them to travel everywhere like quickly. And the question is why? So I think these are kind of first principles how I think about it. There must be all these civilizations. They must be highly developed. And it's extremely likely that nearly none of them makes it beyond a certain point, which is a little scary when you think about it. And that point is, we are not at that point. It's the point where you can easily travel everywhere and see everything, which in theory shouldn't be a problem, right? So my theory is, it's a little dark. Normally I'm very optimistic, but I'm just reasoning here. It's not my opinion. I just reason like what's most likely. And absurdly, it has actually something to do with something very trivial, you would think, and that is terrorism. I think the problem is, if you develop as a civilization, right, there is a certain equation, like how much power does this civilization have? Like the power to change something and to destroy things. It's always the same thing. And of course, there's a totally exponential trajectory. If you go on this trajectory, you have 1 billion times more power in 1,000 years. But that power also applies to each individual in that civilization, right? So if that power is so exponential, it's only a question of time until you reach a technology point where every single person in that civilization becomes capable of destroying the entire civilization. Like it's just a question of time. And we think about atomic weapons and bioweapons. You take this times 1 billion, even if only 10% of the population would be able to destroy the entire civilization, you turn into that scenario where you have to rely on every single human being not to decide today to kill everyone. And I think that's an impossible equation. At some point, if you have a trillion people at some point, and every single one of them at every minute could destroy all others, what are the odds that this never happens? Like at some point it becomes impossible. And so I think exponential and everyone talks about AI in the end, I think that's a much more severe problem. Like at some point, your power exponential curve and the distribution, the inevitable distribution of power leads to a scenario where you can't survive.
Listen, I don't think that you're very far off, right? Which is kind of the scary part of a lot of this, in that the technology that we have, access to as individuals today, right? People always talk about, you have a supercomputer in your pocket compared to, the 1950s or 60s, right? So that's only, what, if you kind of look at it, it's really only 60 years of progress, 70 years of progress. Seems like a lifetime, right? For one human, but on the grand scale of human progress, it's a blip. So it's,
yeah, no, if you just, I mean, here we are talking about why these civilizations don't exist and how they might get, extinct. If you take atomic bombs or hydrogen bombs and compare them to whatever happened before, what was the biggest weapon before? Like some cannon or a machine gun or something in like 1890. So then you have an atomic bomb. What's the delta? It's pretty severe. And now just go on another 200 years. If that same leap happens again on a log scale, the next weapon category then already becomes a little shaky, the whole thing. And then in another 200 years, you have another log scale. So, you have something that's like 10 million times stronger than like a big nuke. So then you can already like wipe out the planet. And then AI on top of that. So I think it's inevitable that you generate more and more of these technologies that are extremely awesome and powerful, but the margin for error becomes just slimmer and slimmer, like exponentially slimmer. If anything goes wrong, then all of us are gone. And I guarantee you in 1,000 years or 2,000 years, I mean, there's no way around that little problem. There might be a solution to the problem, but it's a very complicated solution, I assume.
Yeah, for sure. To wrap up, you get to ask me one question. What one question do you have?
What do you enjoy most about your podcast and talking to different people?
Oh, this is the most selfish thing I do. I get to talk to some of the smartest people in the world and ask them all the questions that I have. Right. And yeah, I record the conversations. Everyone else can listen, but for me, it's just, I get to learn. And by far that's the most rewarding and enjoyable part of it is, I learned things today. I learned things yesterday. I'll learn things tomorrow. And it's pretty cool that, you would take an hour out of your time to sit down and kind of answer questions and teach me things. And so I'll keep doing it.
How did you get into it? What was your first step? I'm kind of interested. Like, how did you engage on that journey, and what were the main obstacles in the beginning that you faced?
Yeah. There was a couple of guys who came to me and said, "Hey, you should start a podcast." And literally I was like, "Well, what's a podcast?" Cause I thought that I was listening to pre-recorded radio, in the beginning, when I would listen to things like a Joe Rogan, whatever. We recorded 3 episodes and we launched it, and they were a huge part of helping me kind of figure out, how do you launch a podcast and all that kind of stuff. And there was just enough people who paid attention to those first 3 episodes where I said, well, maybe I should do a 4th or a 5th. And then kind of from there, it's one of these things where you just kind of put one foot in front of the other, right? And next thing you know, you turn around and you're like, oh, I've done over 300 of these now. Like, that's kind of a lot, right? And so it's been fun, and I'll keep doing it until one day I'm going to wake up and say, all right, no more fun. I'm done. Like, I'll just walk away. So I don't know when that's coming, but I don't think anytime soon.
Awesome. Yeah. Thanks a lot for doing that. I always I admire people who build these podcasts because I learned a lot from,
For sure. Where can we send people so that they can learn more about you and Quantgene?
ChooseSerenity.com. That's our product site. So ChooseSerenity.com, or directly to Quantgene.com. And people can also reach me at jb@quantgene.com if they want to get in touch directly.
Got it. So JB at Quantgene, and we'll put some links in the description for you as well. But listen, Joe, I really appreciate you doing this. This is super fun. I learned a ton, and I hope everybody else does as well.
Awesome. Thanks so much, Anthony. I really appreciate it.